Good morning, everyone, or good evening, wherever you're at. My name is Mahita Chava, and I'm here today to present my research paper about the immunosuppressive landscape of primary versus recurrent brain tumors. Okay. So first, a little bit of an introduction to who I am. So as mentioned, my name is Mahita Chava, and I'm presenting from San Ramon, California, which is about an hour away from San Francisco. I'm a junior attending California high school, where some of my favorite subjects are AP Bio and Human Body Systems. In my free time, I love to play tennis, watch movies, and hang out with friends and family. And before I get into my presentation, I would like to thank my mentor, Shashwat, who guided me through this process and gave me all the resources I needed to successfully complete my paper. So a little bit about the paper before we move on. So like mentioned before, my research paper focuses on the immune suppressive landscape in primary and recurrent gliomas. And this topic particularly interested me because I always wanted to do something in cancer research, but my mentor kind of helped me realize that I was interested in the immune suppressive landscape in tumors. So the overall goal of this paper is to analyze the presence of immune suppressive markers in primary and recurrent gliomas. With this paper, further questions can be asked, such as what does this mean for the patient's immune system, and how do cancer interventions allow the immune system to fight off cancer? Next, I wanted to talk about a little bit of background information, so kind of key terms and key concepts that need to be understood for the overall understanding of this paper. So the first thing I wanted to mention were the hallmarks of cancer. So here, the hallmarks of cancer are basically traits that scientists have observed in pretty much all cancers. These are biological capabilities that a tumor obtains as it grows in the human body. So in the image to the left, you see the hallmarks of cancer in 2010. There were 6 identified by scientists, but this has grown to 14 in 2024. So within this paper, we will be looking at a couple specific hallmarks of cancer, including avoiding immune destruction, tumor-promoting inflammation, and activating invasion and metastasis. So one key thing to note is that most cancers, especially metastatic ones, the tumor cells are able to turn off signaling, which allows them to evade the immune system and grow throughout the body. The next thing I wanted to talk about were some keywords. The first is a glioma. So a glioma is defined as a brain tumor, and high-grade gliomas are malignant with a median survival under 16 months, and they often originate from the glial cells of the brain. The next thing I want to talk about was the difference between a primary tumor and a recurrent tumor, which is really key to understand the overall goal of the paper. So a primary tumor is defined as the original or first tumor present in the body. Most patients often don't die, or their health doesn't completely deteriorate from the primary tumor due to the multitude of cancer interventions that are available, including surgical resection, chemotherapy, and radiation. The next kind of tumor is a recurrent tumor, and this is defined as any kind of cancer or tumor that comes back after a period of time when it was undetectable. And it is often the recurrent tumor that deteriorates the patient's health and ultimately leads to their death. And it's also important to note that a recurrent tumor can often metastasize. So, and the last thing I want to talk about was an immunosuppressive marker. An immunosuppressive marker is a substance that is associated with the tumor microenvironment that contributes to the inability of the immune system to recognize and attack cancer cells. And this kind of just ties back to the hallmark of cancer that I mentioned earlier, where they can kind of turn off signaling to evade the immune system. Next, I want to talk about a little bit of the methods that I took. In a really small nutshell, I collected data, analyzed p-values, analyzed medians, and did further research on certain immune suppressive markers. Going in depth into each of these steps. First, I collected data. So when I collected data, I used the data from the GlioVis website. And I originally started with a list of 32 immunosuppressive markers and immune activation markers. So before I mention the research I did on p-values, I wanted to mention that a p-value is a statistical value that tells you how much the presence of the marker has changed between the primary and recurrent tumors. So if the p-value is less than 0.05, it implies that change in the presence of the marker is significant. So after looking at the p-values of all these 32 markers, I decided to narrow in my research on the immunosuppressive markers because the trend for immune activation markers was pretty consistent in gliomas. So the next step was to analyze p-values. So I already mentioned what p-values are. So by analyzing p-values, I noted down which immune suppressive markers had p-values less than 0.05, so I could do further research on them. The next step was to analyze medians. So by analyzing medians, so essentially the p-value is a change in median between primary and recurrent tumors. So the next step I took was analyzing how much the median changed between primary and recurrent tumors. Noting these down, I chose to do further research on the immune suppressive markers whose median changed a bit more than others. These included galcitin-3, OPN, TGF-beta, and B7H3. The further research I did on these markers included looking at the overall function as an immune suppressive marker, their role in gliomas, and possible clinical trials that could be ongoing to develop inhibitors for these markers. Results. So here is a very kind of summarized result of what I got from researching these molecules or these markers even further. So for OPN, we see that the median increased by 0.46, making it one of the markers I decided to do further research on. In gliomas, OPN blocks activation-induced cell death of macrophages and T cells and promotes the metastasis of the tumor. In TGF-beta, the median increased by 0.26. When researching the drug more, I found out this immune suppressive marker also takes on roles as an immune activation marker. So as an immune activation marker, TGF-beta suppresses and inhibits the function of immune cells, but as an immune activation marker, it regulates stem cell differentiation amongst other functions. The next marker I wanted to talk about was B7H3. B7H3's median increased by 0.39, and as an immune suppressive marker, it inhibits the tumor-specific antigen response and supports many of the hallmarks of cancer, including angiogenesis and the metastasis and migration of the tumor. Lastly, galcitin-3's median increased by 0.77 from the primary to the recurrent tumor. Galcitin-3 promotes the metastatic nature of the tumor, suppresses the patient's immune system to kill the cancer cells, and also provides the cancer cells with the ability to be resistant to drug-induced cell death. Like TGF-beta, galcitin-3 also exhibits natures of an immune activation marker. So one of the major trends you will see with these results is that all of these immune suppressive markers exhibit a lot of the hallmarks of cancer that allow the tumor to grow and invade other parts of the body. I also wanted to mention that while the difference between the primary and recurrent median seemed rather low, it was higher compared to the other markers where the differences were like 0.03, 0.04. Next are some box plots that I constructed on the Excel spreadsheet to kind of show you the visual representation of how the presence of the markers change from the primary to the recurrent tumors. So all these box and whisker plots do look the same. If you look really closely at the median line is slightly higher in the recurrent tumors than the primary tumors. If I'd shown you the same graph for any of the other markers, the median line would have been the same for both the primary and recurrent tumors, indicating a very minimal or if not no change between the primary and recurrent tumor. Interventions in clinical trials. So for all of these 4 markers, clinical trials are underway to develop inhibitors to block the function of these markers. So for OPN, there's a monoclonal antibody that has been developed by Pfizer to inhibit the OPN marker. But since the development of this inhibitor, the manufacturing has since been halted and has not been FDA approved. TGF-beta actually has 2 inhibitors in the making, and the reason for this is that TGF-beta is one of the heavily researched immune suppressive markers, and there's been a lot of papers and a lot of research being published about this marker. The first inhibitor is LY3022859, which is also a monoclonal antibody that blocks the binding of TGF-beta. The study of this antibody has been halted as it was deemed unsafe. The second inhibitor is also a monoclonal antibody called frezolumab that neutralizes the function of TGF-beta 1, 2, and 3. It has passed phase 1 of clinical trials in melanoma and renal carcinoma, and it is currently in phase 2 of clinical trials in lung cancer. So the inhibitors for B7H3 are actually kind of unique because it's not actually an inhibitor, it's actually a therapy. So the most researched inhibitor for B7H3 would be CAR T cell therapy. And for background information, CAR T cell therapy is the idea of engineering and strengthening the patient's own T cells and injecting it back into the patient's body, hoping that it will recognize and kill cancer cells. This kind of treatment is especially common for leukemia and other kinds of liquid-based cancers, and it doesn't work as well for solid cancers. So far, CAR T cell therapy has demonstrated accepted tolerance and promising effectiveness, and it can directly target cancer cells, which is really helpful in killing the tumor. And then for galcitin-3, there's also a monoclonal antibody, but it's not completely there because there's no definitive results. So for the conclusions, I saw that patients with gliomas have an immune system that is unable to recognize and destroy cancer cells due to the presence of immune suppressive markers. One way the researchers are trying to build up the patient's immune system is to develop inhibitors to block the negative impact of these markers. And further research that can be done, kind of branching off of my topic, is how to improve chemotherapy for patients and what is further research that can be going into for clinical trials for inhibitors against immune suppressive markers. And that's the end of my presentation. Thank you.
Thank you, Mahita. Super exciting to see all the different, therapies that are being developed. I'm curious, in your research, did you happen to come across any stats or any findings about how often is it that development starts and then is halted for these?
So for starting, so most of these clinical studies are often done in like the labs of colleges, so more or less in like the medical environment, but more in the research aspect of colleges. And there are always new like starting points for these clinical studies, but they're more than being successful, they're often halted because one, for the lack of resources and lack of money, and then two, it's also because like a lot of it kind of is based on research of these like students, and they don't have enough information to kind of continue the study for it to be safe. And it's also kind of coming down to how does the clinical study of the inhibitor affect other parts of the body, because you don't want to cause the detrimental parts of the patient. You don't want the patient's health to go down just because this seems to be targeting something that works. So yeah.
Absolutely. Gosh, it must take a lot of perseverance to just keep trying when,
Yeah.
so many of them end up not working out. Well,
Yeah.
thank you so much for sharing.